we're going to be talking about the structure of proteins. So what are the functions of proteins? What is the structure of an amino acid? And how are dipeptides formed? So, functions of proteins. So they can be enzymes, antibodies, channel proteins, or structural proteins. So enzymes are globular proteins. They are roughly spherical and they are involved in things like metabolism, digestion, etc. And they have this very specific 3D shape because obviously the active site is specific to a certain substrate and they are complementary shapes. Next we have antibodies. Now you do look at these in more detail later on in the module, but I'll just briefly go through the structure. So you have two heavy chains, these are the blue chains, two heavy long chains and two short light chains. The pink ones are short light chains. Then the darker bit here, you can see the dark blue and the dark pink are called the constant region. And they're called the constant region because they are the same in every antibody. So they have the same amino acid sequence in every antibody. So they're the same for every antibody. And the lighter bits here, the light blue and light pink, are called the variable regions. And the reason they're called the variable regions is because they have a different amino acid structure, and that's what makes them specific. So they have a different amino acid sequence in that variable region, depending on what antigen that antibody binds to. And there are two binding sites, so it can actually bind to more than one antigen, which helps with its function of clumping together those antigens or pathogens, etc. So that's antibodies. Got transport proteins such as channel proteins. So these, so you can see here in pink, they help to get polar molecules through the cell surface membrane, because as you know, the phospholipid bilayer, the phosphate heads are hydrophilic and the fatty acid chains are hydrophobic, and therefore they form that bilayer. So it's to get molecules through that phospholipid bilayer. And then finally you've got structural proteins such as those found, keratin, found in nails and hair, and also collagen. So the reason keratin is so strong is because it has cross-links which stops the fibers from sliding past each other. So they're very strong structural proteins. So the four functions are the enzymes, antibodies, transport proteins such as channel proteins, and you've got your structural proteins as well. So if we now look at the structure of proteins, the monomers of proteins are called amino acids, and you need to know the amino acid structure, the general amino acid structure, from memory for your exam. So the structure of proteins are, you have a central carbon, and then next to that you have an amine or amino group, which is an NH2. On the other side you have a carboxyl group, which is a C double bond O OH, and then you also have, so your carboxyl group is C double bond O OH, so your Q group or your carboxyl group. You have a H on the central carbon, and then your R group is your variant. So this is what is different between each amino acid. And the 20 different types of amino acids that you can possibly have, and it is the R group that is different between each of these amino acids. This bit here is always the same, this bit here is standard. So just to go through that again, you've got your central carbon, which is bound to a hydrogen, and then your R group, which can be different between every amino acid. You've got your amino group, which is NH2, and then on the other side you've got your carboxyl group, which is C double bond O OH.